Okay, welcome to Medicinal Chemistry. And this is Monday, March 30th. And we'll spend about 20 minutes talking about chapter 15. So we're going to talk about how to getting the drug to market. And your quiz, this week's quiz, is on Canvas. Okay, so you're going to have to log on to Canvas on Wednesday for your quiz. And you have 10 multiple choice questions on Canvas, and they pertain to chapter 15. You also have your last POGL participation points. Some people already have all 13, eight assignments of 13 points. So if you haven't gotten your POGLs in, you might want to do that because that is a lot of your participation points, all of them. And your last POGL is over chapters 15 through 18, and you'll see this material on Quizam 4. And then your final exam will cover Quizam material 1 through 4. So there won't be any new material, and you'll use those as study guides. Okay, so let's go over chapter 15 and the concepts. So we're talking about getting a drug to market. So you've already done some of the processes, like you've identified last chapter's Quizam, you identify a target disease, you identify a drug target, you establish the bioassays in order to evaluate your compound. As a medicinal chemist, you found a lead compound, and then you did structure activity relationships because you wanted to optimize that. You identified through that process the pharmacophore, which is the required minimum structural parts of the molecule that are important for bioactivity. And then we've talked about drug design. So you were able to optimize the target interactions. So you thought about whether you needed hydrogen bond donors versus hydrogen bond acceptors. And then we've also talked about optimizing pharmacokinetic properties to minimize phase 1 and phase 2 metabolism. So you've talked about structural changes. So hopefully you feel confident about that. And now the last process of a drug design, let's say you've done all the optimization and now you want to get that drug to the market. So you have to deal with preclinical trials. Okay, so you're going to have preclinical trials. You're going to have phase 1 clinical trials, and you're going to have phase 2, and you're going to have phase 3. And some people even call phase 4 clinical trials, and that's post-market. So you need to know each different specific aims and the descriptions of each one of those. And then we'll talk about patenting and regulatory affairs. And these happen together, okay, these clinical trials and also the patenting and regulation. Okay, so in preclinical trials, the important specific aims or goals, you have to identify the drug metabolites. And you test in animals. The whole point is you want to determine if there's any toxicity. Okay, you have to determine the properties of drug metabolites. So you're doing in vivo and in vitro test for toxicity. So toxicity is a big deal for preclinical trials. Also the pharmacology, you have to identify the action at the drug target. And you're also doing stability tests. So when you look at your expiration dates or how you store your shelf life, that's been worked out at the preclinical trials. A lot of times you put it in UV light and you see the degradation of your molecules. You might put it under acid or base conditions to find the different storage, humidity. A lot of times you use humidity chambers. You might put it under accelerated conditions like really hot temperatures like 100 degrees, and you find out you track by HPLC mass spec the metabolite, and you follow that breakdown. And then the other thing you do is method of delivery. So this is where you're going to decide if you're going to have an inhalation drug, if you're going to have an injection, or you're going to have maybe an oral drug. Okay, and that's called formulation. And as a chemist or medicinal chemist, you can study any of these. You can go into the patent law, you can just focus on pharmacology, or you can focus on formulation stability testing. So looking in your book, there are some terms that you need to know and for toxicity, and one of these is ED50, and there's also LD50. LD50 is the lethal dose, okay, required to kill 50% of a group of animals. It's kind of sad, isn't it? So you have to determine the toxicity, and one of those is the LD50. So 50%, what's the dosage required to kill 50% of the animals? Now ED50 is the dose required to produce the desired effect in 50% of the animals. And then you have a so what you're going to determine in your preclinical trials is your therapeutic index or therapeutic ratio. And so that's LD50 to ED50. So like a 10 to 1 would be 10-fold increase in the ED50 dose that would result in 50% of the death rate. So the dose response curve for a drug's therapeutic and lethal effects can be compared to determine whether the therapeutic ratio is safe or not. Ideally, the curve should not overlap the x-axis, which means that the more gradual the two slopes, the riskier the drug will be. And you can see in figure 15.1 a comparison of therapeutic and lethal doses. And so you're going to get that graph. A better measure of the drug safety is to measure the ratio of the lethal dose for 1% of the population to the effective dose for 99% of the population. So a ratio of LD1 to ED99 of 1 would be safer. Let's look at thalidomide as an example. So you know I like to always tell you something and then I like for you to find an example. So let's look at thalidomide. Now thalidomide, and I'd like for you to actually look it up. Wikipedia would be fine and read about it. I know the structure of thalidomide very well because I actually, do I have that? This was I did 2 years of research on thalidomide, and that was my master's degree, and I would make, I got a 5-membered ring here. So thalidomide looks like this. And one of the enantiomers here, right here, so you have one asymmetric site and asymmetric carbon. And one enantiomer works great, okay? What does it do? It prevented nausea, so it decreased nausea in pregnant women. And so the whole world was taking this drug, thalidomide. I want to say in the 50s. You have to look that up, 1950s, 60s. The FDA, the FDA for the United States of America said, no, we're not passing this drug. Okay. About that time, there were these deformities, and they're actually called thalidomide babies. And so the arms would come out of the shoulders. You need to look at these up and look at pictures. And it's because these deformities, what happens is thalidomide is an angiogenesis inhibitor. Angiogenesis inhibitor. And what that means is it stops the blood vessel growth. So if you think about the placenta, what you need is new blood vessel growth constantly. And this is constantly stopping that blood vessel growth. And so what happens is the babies grow with these deformed limbs. And the FDA said no. They still don't know. Scientists still don't know the exact mechanism which thalidomide works. They do know that this point here racemizes, okay, the asymmetric center, and one of the enantiomers is responsible for the angiogenesis inhibition. The other one would be responsible for the anti-nausea, okay? But it racemizes in the body. So when it gets absorbed in the blood and metabolized, it racemizes. So that means you have a 50% R and 50% S. So there's no way, even if you take just one enantiomer of the drug, it's still going to have the same effect. And so now Celgene actually has made a lot of money off of this drug for anti-cancer, but now pregnant people are not allowed to take that drug. Okay, so that's an example of toxicity studies. Now, this is the thing. The toxicity studies were done in dogs. Let's see. So you have to do a large variety of different in vitro and in vivo testing. So you want to do a lot of different in vivo and in vitro testing so you can see a full toxicity. Thalidomide was developed. Nobody appreciated that drugs could cause fatal deformities, and so there was no test for this. Moreover, even if there had been such tests available, only in vivo tests on rabbits could have detected the potential risk. And so they didn't do in vivo tests on the rabbits. And that's why thalidomide got passed. Now, so those are your preclinical goals. It's all about toxicity, right? And the LD50 and the therapeutic ratio. Now before we go on, let's look at the other example. Let's do one more example. This, we might break this into Monday and Wednesday's lecture. Okay, Monday and Wednesday for chapter. We use both days to cover chapter 15, and you might want to watch that before you actually take your quiz. Okay, so let's look at the drug molecule. This drug molecule here. And you probably recognize this structure as that DNA or RNA. Looks like maybe RNA. There's no OH there. Okay. So this is called a fluoridine. Okay, and there's your iodine there. Okay. Now this was an antiviral agent, and it passed the toxicity test. in the preclinical trials on animals. Okay. However, the clinical trials had to stop when the drug caused severe liver and kidney failure. Now, this is a sad thing. Half the patients, there's 15 patients total, half, 50%, suffered liver failure. 5 people died. Okay, and with two survivors had to have liver transplants. So it was later found that the drug was incorporated into the mitochondria DNA, something that was not observed in the animal toxicity test. So that is the goal of your toxicity test, is just to really understand in the preclinical trials is whether it's toxic before you go into the human trials. Okay, so let's go to the next pages. We're going to look at page 286 to 289. Okay, we're going to cover that to 290. We're going to cover these different clinical trials. Okay, so let's look at phase 1. And if you look at your solvents or your slides, you get a lot of slides talking about how to scale up a reaction, how process chemists look at the different types of chemistry. There's convergent synthesis, there's linear synthesis, and it's all about figuring out the specifications, what your goals are, so you know it's called impurity profiling. You really need to know all your impurities, all your metabolites. You need to know what causes those problems in your metabolism and your animal studies. So it's all preclinical trials. You have to design your manufacturing synthetic process route. You can't change this once you figure this out. This is part of your new drug application, so that's going to be more of the patent stuff. And so as soon as you figure your useful drug and you identify it, you have to carry these studies, these toxicity studies, and figure out how you're going to synthesize the drug before preclinical and clinical trials. You do lose several years of patent protection. Okay, so now we're going into phase 1. So let's say you have your drug molecule and it passed all the toxicity studies. Now phase 1, you're going to get into humans, okay? And they're going to be healthy humans. And you want to establish your dose levels. And you can still, this might be helpful to understand pharmacokinetics and drug metabolism. So now this is the first time you're in humans. So it's usually phase 1 takes about 1 year. Okay. And so when they look at 1990 to 2002, there was an average failure rate of 90% for all drugs that reached clinical trials. Clinical trials involve the testing of drugs on volunteers and patients, so the procedures involved must be ethical and beyond reproach. For all the clinical trials, 1, 2, and 3 can take 5 to 7 years to carry out. It can include hundreds of thousands of patients. It can be extremely expensive. There's actually 4 phases of clinical trials for a drug. So phase 1 takes about 1 year, and usually you have about 100 to 200 volunteers, and they're healthy humans. And the drug is going to be tested on all kinds of dose levels. So you're going to establish your dose level. 6 to 12 subjects are given an active drug. 2 to 4 subjects would be given placebo. During the study, volunteers do not take medication, caffeine, alcohol, or cigarettes so they can avoid complications for drug-to-drug interaction. You can do different special age groups and bioavailability. Remember bioavailability? F is the fraction of the administered drug that reaches the blood supply in a set period. This will be measured. That's your PK and drug metabolism. You'll also want to look for bioequivalence studies. Bioequivalence studies are required when different dosage forms are used in the early and late phase of clinical trials. You might also figure out if you're going to use powder. These are the formulations, and if those different formulations, whether you make a powder capsule drug versus a tablet, that would be establishing your bioequivalence. In situations where the drug is potentially toxic and can be used in a life-threatening disease such as AIDS or cancer, volunteer patients are used for phase 1 studies rather than healthy volunteers. So in an extreme case, you can use unhealthy volunteers. And that's if you need to hurry up and get it to market. I got a feeling they're going to do that with the coronavirus, or they are doing that with the coronavirus. So in phase 1, they're actually using COVID-19 infected patients. Okay, phase 2, clinical trials. They generally last 2 years. This is 2 years. Then you got 1 year, phase 1, 2 years for phase 2. They usually start before phase 1s are complete, so there's some overlapping on each one of these. And you can divide phase 2 into early and late stage. So they phase 2A and 2B. They're usually a double blind, placebo controlled. So even the doctors that are in charge of the clinical trials don't know which patients are receiving the medication versus which ones are receiving the placebo. Different dosing levels and regimes are determined. So they're determining what's the dosage and how often. And the goal here is to establish if there's a therapeutic effect. Okay, so we went from trying to figure out if things are toxic to just figuring out the dosage in the humans, phase 1, making sure it's not toxic in humans. And then phase 2, you want to establish a dosing regime, how often, and you want to identify side effects. You typically have more patients. The endpoint is the measure that is used to determine whether a drug is successful or not. It can be any parameter that is relevant, measurable, sensitive, and ethically acceptable. Okay, these endpoints could be blood assays, blood pressure, tumor regression, disappearance of invading pathogen from the tissues of the blood. So you'd establish what your therapeutic effect is by your endpoint. I know when we did cancer, prostate cancer, an effective prostate cancer drug decreased the tumor size and extended life by 6 months. Okay. And then phase 3 clinical trials. It's carried out by a larger number of patients. And this is going for statistics. They want statistical proof of efficacy. So if it's effective and safety. Okay, and these take usually 3 years. Take up to 3 years, and they're gonna do phase 3A and phase 3B. So once you're on 2B, you can cross over to 3A, and then 3B can go into phase 4. By that time, you already have your dosing, you have your regime, and an example of a drug, Pfizer's torcetrapib, is a cholesterol-lowering drug, and it was terminated in 2006 in phase 3 clinical trials because it was discovered to have a statistically, okay, lots of numbers, increased risk of death associated with its use. The drug had been developed over a period of 16 years, and it cost $800 million and presented one of the costliest failures in pharmaceutical history. And then phase 4, this is just continued. So I actually have a friend that monitors phase 4 clinical trials. She does really well. She works from home, and she monitors patients and the data. But this is continued after the drug reaches the market. So the difference is your drug is in the market, and you still have to deal with your statistics. Okay, you have to monitor it to identify any unusual side effects. Okay, so hopefully you're working your POGL 4, and you have divided up your difference between preclinical trials, phase 1, phase 2, phase 3, and phase 4. And we'll talk about the rest of the chapter 15 on Wednesday.